Hello friends, so in today's video we will study about apoptosis and its mechanism. Apoptosis can be defined as programmed cell death either due to physiological or a pathological condition in which generally affect the small number of cells without showing any inflammatory reactions. Apoptosis in cell can be induced either by intrinsic or extrinsic pathway. To understand in a simple way, let's take an example of Mr. A. Mr. A commits suicide by hanging himself. It can be either due to his personal problems which symbolize intrinsic pathway damaging him from inside or some external pressure due to society or job which symbolize extrinsic pathway. In both conditions, he ultimately dies. Now, let's study about intrinsic pathway. In intrinsic pathway of apoptosis, the death-inducing stimulus is originated inside the target cell itself. This pathway, also called mitochondria-mediated death pathway. Before studying the pathway, first we have to study some basics about BCL2 family protein. The regulatory gene of BCL2 family protein is located on chromosome number 18. BCL2 family protein can be classified into three categories. Pro-apoptotic proteins which promote apoptosis, for example, BAX and BAC. Anti-apoptotic protein which promotes inhibition of apoptosis, for example, BCL2, BCLXL, BCLW. And third category is BS3 only proteins. It promotes or inhibits apoptosis through an indirect mechanism. For example, BID, BAD and BID. In normal and healthy cells, BS3 only proteins are maintained in very low concentration. Anti-apoptotic proteins are able to counter pro-apoptotic proteins inside the cell. But when there is stimulus such as severe genetic damage, hypoxia, high concentration of cytosolic calcium ions or presence of some viral proteins, the level of BS3 only proteins dramatically increased. This increase causes imbalance in pro and anti-apoptosis proteins, causing shift of balance towards anti-apoptosis protein. Now let's see the pathway of intrinsic apoptosis of the cell. Pro-apoptotic factor, for example, BAX, is translocated from the cytosol to outer mitochondrial membrane. It undergoes conformational change and gets inserted into outer mitochondrial membrane, causing pores, that is, protein-lined channels on the outer membrane. It causes increase in permeability of the outer mitochondrial membrane, which is further accelerated by increase in calcium ions. Through these pores, the cytochrome C is released into the cytosol from the mitochondria. The increase in permeability of the membrane causes dramatic loss of electric potential of mitochondria. Therefore, there is release of SMAX, secondary mitochondria-derived activator of capsid, into the cytosol. In cytoplasm, cytochrome C molecule combine together with FF1 and procaspase 9 in ATP-dependent manner, forming a multi-subunit complex called apoptosome. Activation of capsid 9, which further activates the executioner capsids such as capsid 3 and 7, which causes cell to cleave. Extrinsic pathway of apoptosis, the death-inducing signal for the programmed cell death is triggered by external stimulus. The cell possesses plasma memory receptors specific to each stimulus. Therefore, It is also known as receptor-mediated programmed cell death pathway. The external stimuli for the apoptosis in most cases are cytosine, example TNF, tumor necrosis factor. Now let's move to extrinsic pathway mechanism. TNF binds to its receptor called TNFR1 present on plasma membrane, which causes conformational change in that domain. Two cytosolic adapter proteins TRADD and FADD and procaspase 8 residue bind to form a multi-protein complex. TNFR1, TRADD, FADD interact with one another by homologous region present on each protein. Procaspase 8 and FADD possess homologous region called death effector domain in which they interact with each other. Due to this interaction, there is an activation of caspase 8. Activated caspase 8 is an initiator of apoptosis. They activate caspase 3 that carry out the self-destruction process. Hence, extrinsic apoptosis pathway is completed. Another commonly observed extrinsic pathway of apoptosis in humans are by killer lymphocytes through FAS ligand and FAS proteins. The FAS ligands binds to its receptor called FAS on the plasma membrane of the target cell. FAS protein can recruit intracellular adapter proteins that can aggregate procaspase 8 molecule, which helps in activation of caspase 8. The activated caspase 8 further activate the caspase 3 that carry out the cell destruction process of extrinsic apoptosis.